Neurons receive both excitatory and inhibitory synapses. So when fired, each synapse is going to produce a graded potential called a postsynaptic potential that could either be excitatory or inhibitory. So neurons receive both excitatory and inhibitory synapses. So when fired, each synapse produces a graded potential called a postsynaptic potential. That postsynaptic potential could be excitatory, meaning it's trying to elicit an action potential in that postsynaptic neuron, or that postsynaptic potential could be inhibitory, in which case that synapse is trying to prevent that postsynaptic neuron from reaching threshold and eliciting an action potential. So excitatory and inhibitory just refer to if that synapse is trying to bring the postsynaptic neuron closer to threshold, in which case it's excitatory, so it's trying to elicit an action potential, or it's inhibitory, so it's trying to move that postsynaptic neuron farther from threshold and prevent an action potential on that postsynaptic neuron. So graded potentials are also known as postsynaptic potentials. So a graded potential is this brief, localized change in membrane potential. So graded potentials are brief or short-lived, localized changes in membrane potential. Graded potentials typically occur at the cell body or dendrites where a synapse occurs. Graded potentials vary in strength based on the amount of neurotransmitter released at that synapse. So the more neurotransmitter released, the larger that graded potential. The strength of a graded potential is also decremental, which means that it decreases with distance traveled. So the farther away you get from that site of a synapse, the less that graded potential is going to be in strength, or the lower the magnitude of that graded potential, the farther away from that site of stimulus. So graded potentials are brief, localized changes in membrane potential. They vary in strength depending on the amount of neurotransmitter released, and their strength can vary based on distance. And so the current is going to decrease, or the strength is going to decrease with distance traveled, which means that the graded potentials are decremental or decrease with distance. There are two types of graded potentials. There's excitatory graded potentials or inhibitory graded potentials. In an excitatory graded potential, or an excitatory postsynaptic potential, or EPSP, this is a postsynaptic potential that is trying to bring that postsynaptic neuron closer to threshold and elicit an action potential in the postsynaptic neuron. In an inhibitory postsynaptic potential, or an IPSP, this is a postsynaptic potential that's trying to prevent an action potential on that postsynaptic neuron. So it's actually bringing that postsynaptic neuron farther away from threshold in order to prevent an action potential. So an excitatory postsynaptic potential, or an EPSP, is a local depolarization of the postsynaptic membrane. So a depolarization means we're making that cell more positive or less negative. So an EPSP is a local depolarization of the postsynaptic membrane. So in other words, that postsynaptic membrane is becoming more positive or less negative. This is bringing that postsynaptic neuron closer to threshold of about -55. So in an excitatory postsynaptic potential, or an EPSP, we're trying to bring that neuron closer to that threshold of -55. An EPSP is causing a local depolarization because the neurotransmitter that's diffusing from the presynaptic neuron to the postsynaptic neuron is going to bind to chemically gated ion channels that allow for sodium and potassium to pass simultaneously into and out of the postsynaptic neuron. So the neurotransmitter binding is going to open chemically gated ion channels, allowing sodium and potassium to pass simultaneously. More sodium enters the postsynaptic neuron than potassium leaves. So because we're gaining more positive ions than we're losing, that postsynaptic neuron is becoming more positive, or it's becoming depolarized, and therefore getting closer to threshold of -55. So an excitatory postsynaptic potential is a local depolarization of the postsynaptic membrane. It's bringing that neuron closer to the action potential threshold of -55 because the neurotransmitter binding opens chemically gated ion channels that allow more sodium to enter the postsynaptic neuron than potassium leaves. So that's what's getting that cell to become more positive or less negative and getting us closer to that threshold of -55. In an inhibitory postsynaptic potential, or an IPSP is a local hyperpolarization of the postsynaptic membrane. So hyperpolarization means we're overshooting resting membrane potential, or we're becoming more negative than our resting membrane potential of -70. So an IPSP is a local hyperpolarization of the postsynaptic membrane. So the postsynaptic neuron is being driven away from the action potential threshold of -55. So in other words, this neuron is becoming more negative than our resting membrane potential of -70. This occurs because the neurotransmitter binding opens chemically gated ion channels that are permeable to either potassium or chloride. If the channel is permeable to potassium, it's causing the loss of potassium, or the loss of positive ions. When we're losing positive ions, that causes that postsynaptic neuron to become more negative. The chemically gated ion channel could also be permeable to chloride, in which case we would be gaining chloride, so chloride would be entering those postsynaptic neurons. When we're gaining a negatively charged ion, that's then causing that postsynaptic neuron to become more negative. So an inhibitory postsynaptic potential, or an IPSP, is a local hyperpolarization of the postsynaptic membrane. It's driving that neuron away from that action potential threshold, so it's becoming more negative than the resting membrane potential. This occurs because of either the loss of potassium or the gain of chloride. In either case, it's making that postsynaptic neuron more negative. So therefore, we're moving farther away from the threshold of -55. So whether a postsynaptic neuron fires an action potential is going to be dependent on the input to that neuron from one moment to the next. In other words, if you have a bunch of neurons synapsing on one single neuron, there must be more EPSPs than IPSPs. So at any given moment, there must be more excitatory postsynaptic potentials than inhibitory postsynaptic potentials. If there are more EPSPs than IPSPs, and those EPSPs are large enough in strength, it can then cause that postsynaptic neuron to reach threshold at the axon hillock, and therefore cause an action potential. So EPSPs and IPSPs are going to summate in order to determine if an action potential occurs on the postsynaptic neuron. So let's take a look at this diagram up here. So first, in panel A, we have no summation occurring. So here we have a presynaptic neuron sending an excitatory impulse to this postsynaptic neuron. If two excitatory stimuli are sent to that postsynaptic neuron separated in time, so if two stimuli are separated in time, that then does not cause EPSPs to summate or be added together. So if there's enough time in between those two individual stimuli, EPSPs are not going to summate. However, if two excitatory stimuli close in time arrive at that postsynaptic neuron, that then would cause those EPSPs to summate or be added together. So if you increase the frequency of excitatory stimuli, that then causes those EPSPs to be added together. This is called temporal summation. So if two stimuli are arriving from the same presynaptic neuron close in time, those two postsynaptic potentials can be added together. So temporal summation in this diagram is showing what's happening when you have two excitatory stimuli close in time arriving at that postsynaptic neuron. But realize that this could also occur with inhibitory postsynaptic potentials. So IPSPs could also summate if you have two inhibitory stimuli close in time, causing those IPSPs to then summate or be added together. Remember that one neuron can also be stimulated by multiple other neurons. So there can be many synapses on a single neuron. So if you have two presynaptic neurons synapsing on this postsynaptic neuron, and both of which are sending excitatory impulses, we have two simultaneous stimuli at different locations are then going to summate or be added together. So in spatial summation, two simultaneous stimuli at different locations cause EPSPs to then be added together. This can also occur with inhibitory stimuli. So if you have two inhibitory stimuli at different locations, those IPSPs produced can also be added together. Spatial summation can also occur when there are both EPSPs and IPSPs occurring at one neuron. So if you have EPSPs and IPSPs arriving from different presynaptic neurons, so at different locations, those EPSPs and IPSPs could be added together. An EPSP and an IPSP could effectively cancel each other out. So that's being shown in panel D. So temporal summation just means that you have two stimuli that are occurring close in time at the same location. So temporal summation, you have two stimuli arriving at the same location, close in time from the same presynaptic neuron. Those two stimuli are going to produce EPSPs or IPSPs that are going to be added together or summate. In spatial summation, we're going to have stimuli that are arriving close in time but at different locations on that postsynaptic neuron. Those EPSPs or IPSPs can then be added together or summate. So all of the EPSPs and IPSPs, or all of the graded potentials on the dendrites and the cell body, are all going to move along the dendrites and the cell body towards the axon hillock. So graded potentials are going to spread, or EPSPs and IPSPs are going to spread along the dendrites and cell body towards the axon hillock. So the axon hillock is known as the integration point because depending on how strong those graded potentials are, and depending on if they're excitatory or inhibitory, the axon hillock has voltage-gated sodium channels that are going to open in response to depolarization. That could potentially then trigger an action potential at that axon hillock. So graded potentials are produced on the dendrites in the cell bodies. Graded potentials, either EPSPs or IPSPs, are going to spread, and they're all going to accumulate at the axon hillock. So this is the integration point. Those EPSPs and IPSPs are going to be added together. If when all of those EPSPs and IPSPs are added together, we reach threshold at the axon hillock, that generates that positive feedback cycle where we have voltage-gated sodium channels opening, causing that surge in sodium and the resulting depolarization, which is the first phase of an action potential. So an action potential would then begin at that axon hillock. It's important to realize that the distance a synapse is from the axon hillock will ultimately affect its influence. So in other words, if I have an EPSP or an excitatory postsynaptic potential occurring way up here versus right here, the one that occurs closest to the axon hillock is going to have a higher likelihood of getting that axon hillock to threshold, because remember, graded potentials decrease in strength with distance. So the graded potential way up here, or that EPSP way up here, might not even reach that axon hillock. Whereas if I have a synapse right here, the EPSP right here only has a short distance to travel to reach that axon hillock. So that means it's going to be more likely to actually cause that axon hillock to reach threshold and generate an action potential. I also mentioned that graded potentials can differ in strength, so we can vary the strength of a graded potential, an EPSP or an IPSP. Synaptic plasticity, or synaptic potentiation, is one way that we can change the strength of an EPSP. So repeated or continuous use of a synapse is going to lead to a greater release of neurotransmitter at that synapse, and therefore a greater EPSP. So synaptic plasticity, or synaptic potentiation, is when you have repeated or continuous use of a synapse, leading to increased release of neurotransmitter at that synapse, and therefore producing a greater EPSP or a stronger EPSP. So in other words, we're bringing that postsynaptic neuron even closer to threshold. That means that it's more likely that that postsynaptic neuron is going to fire an action potential. This is what occurs in long-term memory. So long-term potentiation in the hippocampus functions in learning and memory. So the more that you use a neuronal pathway, the more likely that you're going to be able to remember something, right? So the more times that you repeat some concept that you're trying to study, the more likely you're going to release more neurotransmitter, the more likely you're going to produce a greater, or you're going to produce a larger EPSP, and therefore the more likely that postsynaptic neuron is going to fire and you're going to remember that concept. So synaptic plasticity, or synaptic potentiation, occurs in long-term memory. So by using your neuronal pathways, you're essentially making them stronger because you're producing larger EPSPs and therefore increasing the likelihood that that neuronal pathway is going to fire those action potentials and allow you to remember that concept.